Alright, this video is going to be the last one for part 1 of chapter 12. It's going to be objective 4. And we're going to take a look at the proteins that are in the membrane of the neuron. And so these are going to be really important because when we get to part 2 of the nervous system, we're going to be talking about action potentials. And so we kind of really need to understand what the membrane is like and the movement of ions across that membrane. And so that's what we're going to look at specifically here, are these channel proteins that are going to be moving ions across. So the nervous system does have 3 major functions that we kind of mentioned earlier. So we have sensation, where it's taking in sensory information. We have integration, where it's processing that information and deciding what to do with it. And then we have the response, where the body is going to do something. A muscle is going to move, a hormone is going to be released, whatever it might be. And so the neurons are able to accomplish this function because they are excitable. They have these membranes that are able to generate the action potentials. And that's just going to be an electrical signal that's moved along the neuron. Now, in order to fully understand what an action potential is, we have to understand what's called membrane potential. And we have to understand how ions are going to move. And just remember that an ion is a charged particle. And so they're going to carry like a positive charge or a negative charge. And we're going to look at the movement across the membrane of that neuron. And so what is membrane potential? Okay, and this really applies to any cell. This isn't just a nerve cell. But membrane potential is a difference in charge across the cell membrane. So remember that inside a cell you have the cytoplasm and the cytosol. You have that solution. And there's all different kinds of things that are going to be dissolved in that cytosol. And then we have our extracellular area, which is going to be the solutions and substances that are outside of the cell. And so anytime there's a charge difference between inside and outside the cell, we have what's called membrane potential. And so this membrane potential is measured in millivolts. It's just like electricity is measured in voltage. The membrane potential is measured in voltage. And so what we're looking at is the inside in comparison to the outside. So when we apply a value here, it has to do with the charge inside the cell versus the charge outside the cell. And so essentially the membrane potential value is telling us what the charge is inside, assuming the outside is 0. Okay, so when we look at this value here, the charge inside the membrane would be -70 if the charge outside was 0. Okay, so at rest, and we're going to see this multiple times. Okay, we're going to talk about resting potential again in the next lesson pretty heavily. But at rest, the neuron's membrane potential is a -70 millivolts. And so that just has to do with that charge difference across the membrane. So inside is going to be negative. If it has a negative symbol there, that means the inside is going to be negative in comparison to the outside, which is going to be positive. And so at rest, the ions are distributed across the membrane in a very predictable way. These are kind of must-knows, okay? So sodium is going to be greater outside the cell, and potassium is going to be greater inside the cell. Okay, both of these are just positive ions. This is sodium, this is potassium. And when we talk about ions moving, these are going to be 2 of the main players in our action potential and the movement across the membrane. Now, in addition to these ions, these are not the only ions that are going to be dissolved in the cytosol or the extracellular fluid. And so these are going to be the main kind of characters of sorts, but they're not the only ones that are there. And so in addition to these ions, the cytosol inside the cell, there's a lot of anions, and there's a lot of negatively charged proteins. And so at rest, this is really important here, at rest, the inside of the membrane is more negative, and the outside of the membrane is more positive. Okay? So taking a look at this picture here, okay, this represents the outside of the cell, and this represents the inside of the cell. Remember, a membrane is a phospholipid bilayer, so that's why we see that structure there. Okay, so here we have a neuron. Out here we have a positive charge, and in here we have a negative charge. And the same thing applies to the other side of the membrane. We have negative on the inside and we have positive on the outside. Now, something else we need to understand is that initially outside there is high sodium. So outside the membrane, there's going to be high sodium. Same thing is going to apply on the other side. And your potassium is going to be higher on the inside. All right, so this is essentially what a neuron looks like at rest. You have positive on the outside, negative on the inside, higher sodium on the outside, and higher potassium on the inside. And overall, that's going to give us that resting membrane potential of negative 70 millivolts. All right, so when a neuron is at rest, the sodium ions are in high concentration outside. When a neuron is at rest, the membrane potential is negative 70 millivolts. And when a neuron is at rest, the inside of the cell is negative. Okay, we got to understand the resting potential before we can really understand an action potential. All right, so that resting membrane potential does describe a steady state, okay? It is going to be maintained by ions leaking across and ions being pumped across that membrane. And so in order to generate these action potentials that we keep talking about, the membrane potential has to change. And so we're not going to talk about the process of action potentials too much yet, but we are going to talk about the proteins that are going to be involved in making that change, okay? So both maintaining rest potential and action potentials are going to involve these proteins. Now, these proteins are in the membrane, and the membrane of the axon is known as the axolemma. So anytime you see that, that's just a cell membrane, okay? So this right here is the axolemma. It's the membrane that is going to be on the outside of the axon of the neuron, okay? And once again, we can see that the outside is going to be positive. The inside is going to be negative. Here we have lots of sodium on the outside. Here we have lots of potassium on the inside. All right, so we're going to look at four main types of channels, okay? They are very specific. A protein is always going to be very specific about what it does, and what that means is that these channels are only going to allow certain materials to pass through. Okay, so we're going to take a look at the characteristics of each one, as well as what kinds of these that we have in our membrane. So starting with leak ion channels, okay, number one, they are passive, which means molecules are moving high to low, and this does not require energy. So molecules will naturally move from high concentration to low concentration. Molecules will naturally move down their concentration gradient, okay? So as long as these are open, which they are, if there is a difference in concentration, then these ions will be moving through these channels, okay? So we have two types here in the axolemma. We have the sodium leak channels and we have the potassium leak channels. And there's actually about 3 times more of the potassium than there are sodium. Now, think about the concentrations, okay? Sodium is higher on the outside. All right, so on the outside we have high sodium. On the inside we have high potassium. So in terms of movement, these molecules are going to move from high to low. So sodium, being higher on the outside, is always going to be moving into the cell through these leak channels. And potassium, which is higher on the inside, is always going to be moving outside of the cell. Okay, and there's about 3 times more of those. All right, the next type is voltage-gated ion channels, okay? And so when it says gated, that just means there's going to have to be some type of stimulus or trigger that's going to open up the gate. In this case, it's going to be a change in voltage, okay? So in order for the channel to be open, the voltage around it has to change. Now, once again, we see passive transport, which means molecules are moving high to low, and no energy is required. So these molecules are naturally going to move through these channels if they're open, okay? We have 3 types in our neurons. We have the sodium voltage-gated channels, the potassium voltage-gated channels, and we also have calcium channels, which we're going to see come into play at the synapse. And so in terms of movement, once again, sodium is higher on the outside. So when these voltage gated channels open, sodium is moving in. Potassium is higher on the inside. So when these channels open, sodium is moving out. And calcium is also going to be like sodium. It's going to be higher on the outside. So when the sodium channels open, they will be moving into the cell. All right, our next type of protein is a ligand-gated ion channel. So once again, it's gated, which means there's going to have to be some kind of stimulus that's going to open up the gate. In this case, it is a ligand. And a ligand is just a chemical messenger. Okay, when we talk about the nervous system, your ligand is going to be a neurotransmitter. Okay, and so these are just chemical signals. And so the way a ligand-gated ion channel works is when the ligand binds, the chemical messenger binds to the channel, it's going to open it up. When the ligand is not bound, that channel is going to be closed. Okay. Now, in terms of movement, once again, we have passive, high to low, no energy required. So if the ligand binds, then the ions are moving through. If the ligand is not bound, then the channel is closed. Okay. Now we only have a ligand-gated sodium channel, and so when it is open, once again, sodium is going to go into the cell. Now this ACH here, acetylcholine, is the main neurotransmitter we're going to be talking about in terms of neurons and muscles. So, but there's other neurotransmitters. It just depends on the pathway that you're in. All right, the last protein that we're going to talk about is the sodium-potassium pump. And anytime you see the word pump, it implies that it is going to be active. And so in active transport, we're moving things against the gradient. We're moving them from where they're lower to where they're higher. Molecules do not naturally like to move that way, so it is going to require energy in the form of ATP. Now, the sodium-potassium pump, we actually talked about that already. You should have already watched a video on it, and there's another video for you to watch. But the way the sodium-potassium pump works is it pumps 3 sodiums out and 2 potassiums in. So keep in mind, remember this is going against the gradient. So it's pumping 3 sodiums out where they're already higher, and it's pumping 2 potassiums in where they are already higher. And we'll see the importance of this and why we need it and what it's actually doing in the next lesson. But for now, just kind of understand the movement. All right, passive transport moves substances from an area of high concentration to an area of low concentration. And the sodium-potassium pump is an example of, okay, we see the word pump, and therefore we know that it is active. All right, when a change in voltage occurs across the axonema, remember that's the cell membrane, this causes blank to open or close. Well, if it's a change in voltage, that is going to affect voltage-gated channels. And then number 4, when the ligand-gated channel opens, so we only had one, okay, the only ligand-gated channel that we have in this particular scenario is going to be sodium, so it's going to be one of these two. When the channel opens, sodium ions are going to enter the cell. Remember that sodium's higher on the outside, and so ligand-gated ion channels are passive, so sodium is going to go in. All right, so you do have some handouts that kind of about the proteins. Maybe you can use those for practice. There's also several videos that you can watch to help you understand about these proteins. And then in addition to that, there's some videos just about the nervous system in general, those divisions, the cells, and so forth. So don't forget to do your activities on Moodle and study for your quiz as well as the lecture test over the nervous system.